right now. Wednesday during class, at the beginning, I'll just kind of recover the topics that are fair game for this exam, but it covers everything, just as a reminder, from molecular biology, including that first lesson, okay, including that first lesson. It includes viruses, and then includes through what we're covering on Wednesday. One of the central themes that's been running through all of this is antibiotic resistance. So if you need to review things like minimal inhibitory concentrations or zones of inhibition, I would take the time to do it. I'm not saying that there is specifically going to be on the exam about that. To be honest, I haven't written the exam yet. But do review some of that material if you feel you need to, okay? It may be important to you when we get to the exam. Okay, what I wanted to start with today was to recap the homework assignment. And the way that I want to do this is I don't want to go through every question unless there are specific questions that you have. So I'd like to give you guys 90 seconds just to look through either your notes or go back through your quiz, and I want you to just come up with which ones do you still have, which of the questions are you still uncertain about? Or which of your answers, even if you got, and I'll be making sure to go back and change those 1.8s to 2s later on, but even if you got a perfect score on it, are there ones that you don't understand why the answer is what it is? Okay, so I'm going to give you a couple of minutes to do that, and then we'll talk about whichever ones you have questions about. Okay. Where are there still questions? And direct me by question number 2 if you can. Devin.
I don't know this question number. I think it's like one of the ones towards the...
Which one was it? Maybe the question, the order might change. So now I forgot that. I set it up to change too, but...
It was why you mainly find gram-negative in bacteria in the large intestine.
Okay. Is it this one? If you stain the bacteria that live in the human intestine, you would expect to find mostly? Okay. So this one is really just based on what we know. And this is based on studies of the human microbiome. What would we expect to find? So this is more kind of just a memorization answer than anything else. There's not much rationale to it. What I will point out is that the most abundant microbes within the large intestine is we find gram-negative bacilli. So Bacteroides is one of the 2 major genus, And then there's a larger class known as the Enterobacteriaceae, which include your E. coli, Klebsiella, some of the foodborne pathogens that we might associate as well. Most of these, the conditions of the large or the large intestine, what are the environmental conditions like? So what's the oxygen content like? Ashley? Yeah, so largely the organisms there, because there's minimal amounts of oxygen, honestly, most of it's been consumed by microorganisms that are metabolizing either in the stomach or the small intestine. Because that oxygen is at very low levels, we find anaerobic metabolism occurring. So whether it's anaerobic fermentation or anaerobic respiration. The types of microorganisms that we would expect to find, or those that are most common, are going to be aerotolerant anaerobes and facultative anaerobes. Now, there are very likely to be many obligate anaerobes there as well. But as you may or may not know, I don't know if we've talked too much about culturing anaerobic anaerobes this semester, or obligate anaerobes, sorry. But obligate anaerobes can be really challenging to grow because even the touch of oxygen for some of them can lead to their death. And so one of the powerful tools in studying the human microbiome right now is that we can just take a gram of feces and sequence every piece of DNA in there. We don't actually have to culture the organism necessarily anymore to understand what's there. And so answers like this one here may change over the next 15 to 20 years as we know more about the human microbiome. But as it stands right now, based on what we know, what we can culture, largely it's gram-negative rods that can perform some sort of anaerobic metabolism.
I think like I know the question on that because like there was gram positive and gram negative in there, so I know like it wasn't sure.
Yeah, and I suppose maybe I need to redefine that question and say the large intestine, because if you were to look at the small intestine, what you might find are a greater proportion of gram-positive rods, more of the lactobacilli. If we take the GI tract as a whole, though, where is the greatest concentration of bacteria? In the large intestine, and that's why I think that answer, the gram-negative, is most fitting. So dependent not only on the location, but if we take a look at the whole system, what we're going to see most commonly would be those gram-negative bacilli. Other questions? Abby.
I don't remember what the answer to question 1 is. In the video lecture, you said that the small or the stomach has low concentration.
Yes.
But then I think the answer is
It is, yeah, that's a great question. And I noticed that after the fact as well. So it is true that the stomach, out of all of those locations, has the smallest amount. I think the measurements that, if I'm remembering correctly, the video shows a chart actually of like concentration and then location on the x-axis. And I'm trying to remember if that concentration is in grams or if it's in a concentration per milliliter. Now this one specifically is referring to grams.
per milliliter.
It is per milliliter there. So that might be the difference. Per milliliter, there might be less than 100 in the stomach, but per gram, we think that typically there is more than that amount in the stomach even. And this is important because we think about the stomach oftentimes as being a sterile, relatively sterile environment because why? What is there that makes this a sterile environment? Ashley? Yeah, it's incredibly acidic. We're talking pH of 1 or 2 in some cases. There are some microorganisms that live there. Some of them may even, and we're kind of talking even outside of bacteria, there may be archaea that live in the stomach as well. But the ones that do, we can consider them likely to be acidophiles. More likely, though, they're acidotolerant. They can likely survive more neutral pH as well as those that are coming into the stomach are not oftentimes naturally residing in the stomach, but are coming in transiently. Okay? There are some exceptions. There are some that will remain for long periods of time, like the cause of ulcers, Helicobacter pylori. And I talked about in the video how Helicobacter, for example, its mechanism to survive that acidic pH condition is to secrete bicarbonate that allows it to kind of neutralize the pH in its general vicinity. And that allows then for this organism to create the ulcers that we associate with stomach ulcerations. Does that help to clarify that one, Ann? Okay. Any others? There is one more of these for Wednesday. I had an issue where I didn't originally set it up for you to take it unlimited times. That's been fixed, so please take it as many times as you want to, okay? And these will remain open, so if you want to use these as study tools, it won't regrade you anymore. At least it shouldn't unless I were to somehow override it, but it should, let me rephrase that. It would grade you on how you perform on it, but it wouldn't change your grade in the grade book. That's what I want to say. So go ahead and feel free to use these some more if you want to. But where we're going to move into today then, bless you, is, leave that, is moving into then how this, well, I wanted to cover a couple of brief things here and talk about the benefits, but then move into the potential downsides in respect to antibiotics again. I throw these couple of ones up here real quick to talk about the benefits that we gain from some of these microorganisms. We could consider these to be mutualistic relationships where what we eat provides energy for the bacteria. In return, their products of their metabolism can be beneficial to us as well. And so you may recall in the slides there's a table that looks at, for example, some bacteria produce vitamins that most of us, or some of us these days, supplement as part of a daily pill. But bacteria can provide those vitamins as they are performing their metabolism. We can get some like the enterococci that produce these short-chain fatty acids. And when we say short-chain fatty acid, to give you a context of what we're talking about short-chain fatty acids and taking a look at what is the effect on the colon. And in particular, what they have observed is that some of these fatty acids seem to actually regulate the growth of the cells that make up that line the colon, so the epithelial cells. The importance of that is that it's thought that these short-chain fatty acids might actually have some indirect contribution to preventing uncontrolled growth, which could lead to colon cancer. Okay, that's one hypothesis right now. Another observation is that these short-chain fatty acids contribute to the junctions in between these epithelial cells. And so the tight junctions, which are proteins that hold cells together to help create this lining of the epithelium of the colon, the short-chain fatty acids help to keep those junction proteins intact and keep those epithelial cells from separating or kind of distancing a little bit from each other. So it reduces the permeability of the colon. And the benefit of that is that microorganisms are less likely to escape from the colon into the bloodstream, which can cause inflammation, which can cause a whole host of other innate and adaptive immune responses. So there are some individuals that think disruption to the production of these short-chain fatty acids are contributing to particular diseases like inflammatory bowel disease or Crohn's disease or some of those other ones that are associated with autoimmunity. Okay, it may actually be our response to the microorganism. The other one is there is some link here between production of these short-chain fatty acids and insulin regulation as well. So there are some that believe type 2 diabetes in part might be caused by a disruption of what we would consider to be a quote-unquote healthy microbiome. Okay. And then there are plenty of neurotransmitters that are thought to be produced by bacteria as well, that are thought to have a role on cell physiology, endocrine system, and neurotransmission as well. So I just point this out as this is a very complex topic right now. We are looking at a very, very small piece of this. But when we talk about microbial communities, we're talking about not only the interactions between the microbes, but when we talk about the human microbiome, we're talking about how our microenvironments, the difference between the stomach, the small intestine, and the large intestine influence microbes, and how those microbes can influence our health as well. So let's talk superinfections. Okay. What is a superinfection? I think I have it listed up there. But what is a superinfection? In your own words. I'll tell you what. Here is a definition of it. What I want you to do is talk to somebody else for a minute and rephrase this in your own words. Okay, so what is a superinfection? And if you have to use an example for it, go ahead and use an example. Hey, Emma.
I just like retook it right now. I already took it before I did it. It's not going to count it as late.
No.
Okay.
It is what it is. I've already seen who did and didn't submit it this morning, so don't worry about that. Yeah, it just puts a little like exclamation point there for me to like regrade it for anybody that does it again, but I usually just ignore them for the most part. Isn't that an awesome picture? This is what I do in my free time, is look for nerdy jokes. That could be an example, yeah. Usually when we think of superinfection, I mean, that is a potential example. Sometimes when we think of them too, it can be you take your course of antibiotics and then you get an infection caused by something else. So like, let's say you've got a cut and you're taking antibiotics to like help that wound heal. If that, what was originally infected by by staph epidermidis now becomes infected by Klebsiella pneumoniae, that's a superinfection. All right, let's go ahead and redefine this. What is a superinfection, Kaylin?
Something like C. diff, where you're trying to treat like a sinus infection or something with antibiotic, and then you get C. diff from it.
Yeah, and that's kind of the classic. That's exactly where we're going to go with it today, and that is a perfectly fine example. So a superinfection, when we think of it most often, we see this often in a clinical setting, where somebody is being treated with an antibiotic to treat an infection. The infection becomes cleared, and then there's this secondary infection that occurs afterwards. So let's say, for example, you have a sinus infection would be a fine one. I was also thinking, like, what if you're taking antibiotics to cure an ulcer, for example? And those antibiotics have to be taken orally. Okay, so you take an oral antibiotic, the ulcer gets cleared, but then all of a sudden it also clears out other bacteria within the gastrointestinal tract, and then you allow for something else to populate the area that wasn't the most prevalent species in the past. Okay, and so that's what we're going to talk about today with C. diff. One of the things to keep in mind here is broad versus narrow spectrum antibiotic. So what is the difference between the two of those again? What is the major difference between a broad and narrow spectrum antibiotic?
like specifically towards like one thing or something as broad might be like a general like
Good.
thing?
Good. So narrow, again, would be toward maybe a particular class. Probably one of the greatest examples is ethenbutol and isoniazid. Those two target mycobacterium and nothing else because it targets the synthesis of mycolic acid. Okay? A broad spectrum example would be the sulfa drugs. It targets folic acid synthesis, which is something that is common in every single microorganism. Okay? So think about the effects that that has. Okay? As we're kind of going through this and take a look at this example. These slides are kind of like these are not in the mini lecture, of course, but these are kind of an extension. I added these on to the end of yours. So, and I'll do the same thing for Wednesdays as well, just so you're aware. But one of the things to point out here is that your natural microbiome, and today we're just talking gastrointestinal tract, what's there keeps pathogens away, okay? And the reason for that is there are a couple of different reasons, but one of them are that these fatty acids that get produced, they actually can be inhibitory to some microorganisms. We've talked about how lactic acid, for example, some organisms are not acid tolerant. They're neutrophiles. And so an acidic condition, for example, can prevent them from even growing. And we'll talk about that again in the context of the urogenital tract on Wednesday. Other examples, competing for nutrients. Some bacteria produce what we call bacteriocins, which are essentially proteins that have antibiotic-like qualities. So some bacteria use these bacteriocins to attack other bacteria. Some might actually activate the immune response or help to regulate the immune response as a way to keep pathogens at bay. And the last point that's not brought up here is what we would call colonization inhibition. And what we mean by this term is that within the gastrointestinal tract, there are bacteria that are attached to the epithelium that make up that gastrointestinal tract. Right now inside of you, not only is there a ton of poop, but there are also bacteria that are lining the intestines. Okay? So think of it kind of like you have mucus on those intestinal linings, but you've also got bacteria there as well. And so you can kind of think of these bacteria as, in some ways, they're a protective coat. All right? By completely colonizing the intestinal lining, what it does is it prevents other bacteria that may come through transiently as we eat. They protect us. They protect us from allowing those bacteria to even attach to the surfaces. Okay. So think of it like a city where every single apartment is occupied, nobody new can move in. We've talked about the broad and narrow spectrum again, but this just kind of points out, again, narrow spectrum, goal is target one specific class if we can, broad, targeting different types. Now, in thinking about this, I want to pause for a minute and give you guys a minute to think about this then. But think about taking an oral antibiotic. And what is the path of an oral antibiotic, or anything we eat, I guess? What is the path of when we eat food or drink a liquid? And I'm looking for responses here. Okay, goes in the mouth, through the esophagus, right? I think so. Not trachea. That would be bad. Into the stomach, incubates there for a while. To the... Where is the stomach? I feel like I'm pointing in the wrong areas. Get Brenda or Debbie in here to do this. But after that, goes to the small intestine, travels through a lot of areas in the small intestine, goes through the large intestine, ultimately 2 routes. Urination defecation. Okay? Or if it doesn't go well with you, the third route is out and at 'em, right? Okay. So think about an antibiotic. Think about what are the downsides of this? Okay, what are the downsides of taking an oral antibiotic? And again, put this into the context of what broad and narrow spectrum antibiotics have. Let's talk, I'll give you a minute just to kind of craft something real quick, but downsides of taking a broad spectrum antibiotic orally. Okay? I'll give you a minute to think about that. Okay, so what effect would does an oral broad spectrum antibiotic have on the microbiome? Ashley?
What you're aiming for it to kill off, but also like some other microbes, and then microbes that have been killed off will have higher concentration than they would have been in a normal environment.
Okay, Meg, do you want to add to that?
Well, then they also kill off like all the good bacteria that's in your body, so then your body needs to
Right, and that's the only thing I would have added is that when you're Ashley, when you're talking about that it's killing the ones you want it to kill, but there's also, I mean, sometimes the term collateral damage gets used here. Antibiotics are not specific to who's good and who's bad. It's who has the target that this antibiotic is intended to inhibit. And so there can be obviously downsides to this. Now, one of the things that some medical professionals have suggested is that with antibiotics, we should go ahead and take a probiotic, for example. So go ahead and, as you're taking that oral antibiotic, make sure you're eating yogurt along with it. What's the idea behind that? Why propose something like that, Kaylin? Yeah. And specifically, what kind of good bacteria are we talking about in a probiotic? What is in yogurt? What is in, well, yeah, yogurt. Lactobacillus, your lactic acid bacteria, and so we'll talk about a couple of them. Lactobacillus and bifidobacterium is the other one that's been proposed. Sometimes they say just take an oral pill if you don't like yogurt. That idea is a bit controversial right now. And the reason why it's controversial, it's not that the idea is bad. It's more so that we don't really know if the bacteria actually survive like this. If you're going into surgery, would you rather use a butcher knife or would you rather use a scalpel to cut somebody open? Okay? So, and yeah, this one's a ruptured appendix as the surgery, okay? So, and this is kind of how we have to think about it a bit, in that, are you just going to go in and who cares how messy it is, you get it done, or do we want to be more tailored with our techniques? That's the idea behind a broad versus narrow spectrum antibiotic. Let's just take a look at, let's kind of reiterate what Ashley and Meg just said. Okay, so they pointed out that, all right, so before the antibiotic, good, healthy gastrointestinal tract, we take antibiotic kills good and bad bacteria, but there are bacteria that are resistant to that condition, right? And those that are resistant now become the most abundant species, or at least with this freedom, there's less competition for nutrients, there's more space to colonize, we lose that colonization inhibition, And now these bacteria that are resistant to that antibiotic become the most plentiful. Is this always going to happen? No. Okay, it's dependent on what's in the gastrointestinal tract at the time you're taking the antibiotic. I put this up here just real quick as an example. It's not only C. diff that's a concern, but it's also infections caused by enterococci. This is just looking at, and I'm not asking you to memorize this by any means, but one of the things I want to point out here is that the ones in the dark brown there are, if I'm not mistaken, these are antibiotics that are particularly well designed against actually anaerobic microorganisms. The ones on the bottom, the light gray, are ones that are targeted, that work really well against aerobic microorganisms and less well with anaerobic. And what they're looking at here is how does this vancomycin-resistant enterococci, how do the numbers of those change per gram of feces in individuals that are being treated with these different antibiotics? And what we find here is that at least with vancomycin-resistant enterococci, or VRE, we find that we can actually break down these antibiotics by whether they target whether they target aerobic or anaerobic microbes. The anaerobic ones seem to increase the concentration of vancomycin-resistant enterococci. And enterococci itself is an anaerobe. It's an aerotolerant anaerobe. And so we find that the types of antibiotics we choose can have an effect on the repopulation of the gastrointestinal tract. So how does this relate to C. diff? I mean, some of you have actual personal experience working with individuals who have had C. diff. The only thing I hear oftentimes is it smells, and everybody, whenever I bring up C. diff, are like, "Oh, it's disgusting." But that's my experience with it. So, what are your experiences with C. diff? When does it occur? When do C. diff infections occur? Ashley?
A lot of times it's
Okay, so often occurs after an antibiotic treatment, or maybe during an antibiotic treatment as well. Haley?
Well, that's what I was just gonna say, most of the residents who we send out to like the hospital being treated for something, they come back, it's like 90% of the time they come back with something.
Age level?
60 to 80.
60 to 80? Same?
That's about what.
Yeah, I think that this, at least what we see is it's more with immunocompromised individuals, which the elderly would fit in that population.
Yeah, it spreads like crazy, you know.
Yeah, and it spreads like crazy too. Why?
Like you have to wear masks and gloves and gowns and.
Right, so there's a lot of personal protective equipment somebody has to wear, right? And the design behind wearing that personal protective equipment is to prevent the spread from one location to the next. It's very easy to pick up C. diff, but there are some inherent qualities of it that allow it to be so easily transmitted and so easily picked up from one person to the next. What is one quality of Erica? Spores. It's a great sporulator. Clostridium as a genus are all sporulating bacteria. And so because of that quality, you may remember we looked back maybe about 4 weeks ago now when we were talking about ways to control microbial growth, and we talked about the paper that discussed triclosan, and then I brought up this other table and I said, okay, here's a study that's been done where they're looking at washing hands versus hand sanitizers, and they looked at the amount of Clostridium spores before and after, and guess what? The hand sanitizers don't do anything. And again, the reason for that is spores aren't affected by hand sanitizers. You have to physically remove them, go through a degerming process, or what are the other ways that we can kill spores? Well, high heat, incinerate, and guess what? Neither of those are going to work on a patient, okay? Disinfection. could work, but we've got to use high concentrations. 10% bleach for 2 minutes is not going to work. UV light probably won't work all that well. And so with infections like C. diff, we have to take a lot of care and concern into how do we actually, one, stop the transmission, but two, then disinfect the room afterwards or sterilize the room as a way to kill those spores. So we've covered this a little bit already, but gram-positive bacillus, spore former, opportunistic infector, in other words, and this is common among superinfections. They are opportunistic pathogens. They are ones that not necessarily are going to cause the initial disease, but when the concentration of resident microbes becomes low enough, that allows for these superinfections to kind of gain traction and now begin to grow, multiply, etc. We've talked about a couple of the features of the antibiotics already and how this can kill the resident microbes. In the case of C. diff, it really creates space. So it's as if, let's go back to the city metaphor for a minute. Let's imagine you've got Madison's, 230,000 people? Let's imagine that takes up every housing space in the city, and then all of a sudden we say, all right, 200,000 or 215,000 of you are now going to leave. And that allows for now those people that are the remaining 15,000 people to expand or spread out. And it allows for, because let's assume the resources are still here, it allows for increase in reproduction. It allows for those people to eat more, et cetera, et cetera. So essentially we're talking about, again, evolution on a micro scale here. Selection with an antibiotic creates an environmental bottleneck that only allows for some organisms to actually survive and ultimately repopulate. And then the last one here, the common treatment for C. diff right now is vancomycin. However, we're finding, at least clinically, we're finding an increase in the percentage of C. diff infections that can't be cleared with vancomycin. Either the population is naturally resistant, or there are kind of like what we saw with the article I had you read about the Pseudomonas infection. The numbers go down for a while, and then you get a rebound of the original infection. Here's just a little quick image showing you the epidemiology of C. diff right now. Taking a look at the rate of infections per 1,000 individuals discharged from hospitals. So this one came from 2014, and they had data through 2011 and made predictions about 2012 and 2013. And this is looking at each quarter of the year, how many cases are occurring per 1,000 people that are discharged from hospitals. You can see 2001 it was about 5. We've almost tripled that number, where about 15 are occurring for each 1,000 people that are discharged. In 2011, responsible for almost 500,000 infections, 29,000 deaths. Okay. And then here are just some of the symptoms. Erica. So I think that, so you're saying if you have C. diff in you already, I think you're asking why aren't you sick if you already have C. diff in you?
I mean, like, if I have C. diff in me, clearly my body right now can control the levels of it. So why can't my body like fight against it if I were to like be working with a resident and get it in my own system? Like, why is it so contagious?
I think the personal protective equipment is in part for you because often in those patients the concentration of C. diff is much higher than what you would have in your gastrointestinal tract. I think that's one piece of it. Okay. We talk oftentimes about an infectious dose. How much of a microbe do you need to have ingested, or how much do you need to cause illness? So for like listeria, it's like you have to have ingested 10 million cells to cause an illness. In 50% of individuals, it's always a kind of a normal distribution of how much it is. In C. diff, probably in you, most of if let's say you have C. diff in your system, most of it is kept at bay, and most of it is probably transient too. If you are ingesting it, the colonization inhibition that's occurring from your natural microbiome is probably preventing that C. diff from ever colonizing the gastrointestinal tract. However, if you're working with patients, the concentration of C. diff in that individual is much higher than you would encounter in your daily life. And so I think that increases the risk that you would get infected as well. So I think that's part of it. I think the other part of it is, why do I ask you guys to wear lab jackets in lab? Exactly, you don't take it outside of you. So if you're asked to gown up when working with a C. diff a patient who has C. diff, my guess is most of the time what they're going to ask you to do afterwards is, even before you exit the room, there is a wastebasket there that with a foot pedal, you take off your stuff, you put it in there, you wash your hands, you leave the room. And that's the goal there, is just to prevent the spread of transmission from one person to the next one. So in some ways, it's kind of like it's an isolation technique to prevent the spread across the hospital. Does that help to answer? Okay. I may be preaching to some people here that already know this information, but I wanted to put this up here, and I'm not asking you to memorize this for the exam by any means, but some people may be interested in this information, so I just put this up here so that it's available to you. I think maybe the one that is kind of important, and this kind of hits a little bit with what you just said, Erica. So most cases are going to occur shortly after antibiotic usage. 25 to 40% may not become symptomatic for actually many weeks afterwards. And that's because it takes time for C. diff to colonize and to grow before you experience the side effects of that infection. In some individuals, you may just be lifelong carriers. Okay? Anybody know the story of Typhoid Mary? What's the story of Typhoid Mary? To those of you who shook your head, yes, you know the story. You know of Typhoid Mary? So Typhoid Mary, 1900s, New York City, was a cook, basically an asymptomatic carrier of Salmonella typhi, which is the bacteria that causes typhoid fever. She never had any effects, but basically she was able to spread or pass along that Salmonella typhi to other individuals, and many of them ended up dying. As a result, she got put on an island in the middle of New York City, was basically forced to live there for the rest of her life. Okay, at about the age of 26, did a 3-year stint, went back into the public, decided to start cooking again. They took her back to the hospital. She lived there from like the age of 40 to the time she died. Okay, so there are some people who can be asymptomatic carriers. We don't quarantine most of them anymore, though. All right, so what does this actually do to the gut? I want to just kind of give you guys a couple of images real quick here. And I know we're almost done with time, but so I'll kind of keep this a little bit fast. But this is just some images from, I think this might be a mouse model where they were looking at exposure to C. diff. So what they're focusing on is this location right here. And what they're trying to say is that at the point at which C. diff colonizes, what you start to find is some disruption to the colon lining, as you can see from one image to the next, but you also get the pseudomembranous colitis. And these little pustules or these little pockets are where the C. diff is located. Okay, and these actually become really challenging to remove then. It's somewhat like a biofilm. It's protective. It protects the C. diff from being destroyed by antibiotics and released from this location. The goal then is to eliminate those pathogens. Okay, and how do we do that with C. diff? And one answer is we treat with antibiotics. But if you're treating with a broad spectrum antibiotic like vancomycin, we're already at this step here. Let's assume this is a colon filled with C. diff. If you treat with a broad spectrum antibiotic, that's going to kill off the C. diff, but what else? It kills off other bacteria as well. It doesn't allow the time for the natural microbiome to restore itself. And so that's part of the reason why we see these rebouts of C. diff infection in individuals that are being treated with antibiotic, is that the gastrointestinal tract never has an opportunity to actually restore itself. And so that's led to this. Eat shit and live. Okay? Or eat poop and live. Poop emoji and live. So the idea here, and this has been probably proposed and actually being used for the past 10 to 15 years. I can actually remember the first time I heard a talk on this, and I was like, that's disgusting. And I was imagining like Austin Powers as I was listening to this talk. But the idea is to restore somebody's natural microbiome by actually providing the bacteria straight to the gut. So instead of treating a C. diff infection with antibiotic, let's treat it with poop. 3 ways that this can be delivered, okay, or 3 ways that have been proposed. And all of these are in different stages of being used. The one, tube up the nose, all the way to the stomach. They just deliver the poop right to the stomach. Okay, or right to the gastrointestinal tract. Route 2, up through the rectum. Okay, enema style basically. Route 3, freeze-dried pill. Spores of bacteria like bacillus delivered to the gastrointestinal tract to repopulate the bacteria. Does it sound disgusting? Absolutely. Does it work? Heck yes, it works. Okay? So let's take a look at this real quick. I want you guys to just take a look at this data here. This is percentage cured without relapse. So these are, this was a study done late 2000s. They took a look at individuals who had received donor feces, and a donor here is not like matching blood or matching for like certain antigens. You basically need to find somebody with like a similar diet as you to repopulate your natural microbiome. So like I eat a lot of candy right now, so I would basically want like the Stay Puft Marshmallow Man to be my fecal donor, okay? And then, so this is first infusion. This is if they had to do numerous infusions, how many were cured without a relapse. And then with vancomycin and vancomycin with a bowel lavage where they're basically cleaning out using like a saline solution to clean out the intestines. And what you can see here is the individuals who receive a fecal transplant, the first time through, there's an 80% cure rate. Those people go home after this fecal transplant and no longer have C. diff, whereas those treated with vancomycin, it's only 30% successful. The remaining 70% get some sort of relapse of infection here. Okay? Does it seem disgusting? Yes, of course it does, because it's poop. I mean, I missed this yesterday. I was out when this happened, but my niece was over at our house. She stepped in dog poop, and her immediate reaction when she steps in dog poop is to vomit. So she stepped in dog poop, and then 2 seconds later vomited in my backyard. Okay, yes, it's disgusting, but is it? For those of you looking for homeopathic cures, there's none better than this one. Okay, it seems to work. And this will continue. This is kind of a cutting edge area of microbiology right now, is trying to understand what is our microbiome, how does it develop, how can we restore it, okay? Yeah, exactly. Abby? Oh, yes, this is being used. And there are, you might go online, go onto YouTube, and you will actually see testimonials from people. There have been articles and stuff done on people who have received these sort of treatments and come back perfectly fine. So, like, they were on death's door with a C. diff infection and are now living a happy, normal life. All right? All right, that's it for today. We'll go ahead and talk about the urogenital and skin on Wednesday.